Hi, I'm Julian Moger from the University of Exeter. Thank you for taking the time to watch my video describing my work on label-free imaging in Planter. So let's start with the motivation behind this work. So the issue of global food security is one of the global challenges that we're facing at the moment. We know there's an increasing demand for food due to an increasing global population and increased life expectancy, compounded by the fact that there's a growing demand for a high protein diet in the developing world. And it's predicted that to meet the global demand for food security, we need to increase the yield by around 70% by 2050, which is now only 30 years away. So we're looking to the agrochemical community to make significant breakthroughs in the efficiency of agrochemicals. 40% of the food at the moment wouldn't exist without agrochemicals, but in order to meet the demand, we're looking for a significant step change in the efficiency of agrochemicals without having any negative impact on the environment. However, if we look at the tools available to plant biologists compared to biomedical researchers, the agrochemical industry is working with some relatively crude tools. So we really need to be looking to biophotonics to see what we can develop to give them the tools that they need to deliver this increase in efficiency. So if we look at optical microscopy for biology, the aim is to reveal the spatial and temporal distribution of biomolecules at the cellular and subcellular scale, whilst producing contrast between the different biomolecules, so we can see which sort of molecules are responsible for which processes and what type of molecules enter and leave cells. Most of the work in biology has typically been done with fluorescence microscopy, where the chemical specificity comes by the ability to selectively tag biomolecules with fluorescence markers that emit fluorescence at different colors. And this is a fantastic technique, but in a lot of cases, it would be very advantageous to move to a label-free alternative that doesn't rely on tagging. So if we look a little bit deeper into fluorescent labels, we can see that the molecular weight of a typical fluorophore is around 350. And if we can compare that to a typical protein, which is around 10 to 100,000, you can see that the size of this dye is insignificant, and it shouldn't really perturb the function of that protein. But if we can compare it to something like a fatty acid or a metabolite or an agrochemical active ingredient, you can see that the size of the fluorescent label is comparable, in a lot of cases bigger than the molecule you want to track. So tagging is going to significantly perturb how the molecules interact and how easily the agrochemicals can penetrate in and out of cells and across the various membranes within the plant. So what I'm interested to do is to see if we can develop a label-free alternative for imaging the movement of these agrochemicals in plants. Raman scattering is a very promising technique that could replace the label-free chemical specificity that we can achieve with fluorescent tags with a purely optical technique based on vibrational spectroscopy. As molecules vibrate, for each bond, the photons see a changing polarization that fluctuates at the specific resonant frequency of each bond. And this results in sidebands shifted exactly by the resonant frequency of the bonds from the excitation peak. And if we plot the spectrum of these peaks, what we get is a series of peaks, and the position of each peak corresponds to the resonant frequency of the particular bonds within that sample. And we can use this to map the chemical composition of cells, for example, with very high precision and extract a huge information about the composition of that sample. So we can think of our Raman scattering spectrum as a chemical fingerprint of the sample. So there's a huge amount of potential to completely do away with the need to tag labels and rely purely on extracting information from the scattered frequencies of light that interact with molecules. But unfortunately, the signal that we get from Raman scattering is about 1,000,000 times weaker than the signal that we would get from a typical fluorescent tag. And this limits the sensitivity of Raman scattering to a few millimolar molar concentrations. And it can require acquisition times of up to 100 seconds to acquire a single spectrum from a single point. So if we want to map out high resolution of images of cells consisting of millions of pixels, it's going to take a very long time unless we use a bigger laser. Obviously, if we use a laser that's 1,000,000 times stronger than that used for fluorescence, it's not going to be compatible with biological samples, and we're just going to completely ablate the sample. So this is where the technique that I'm interested comes in, coherent Raman scattering. In coherent Raman scattering, instead of shining a single incident frequency on the sample, we shine two excitation beams onto the sample, one called the pump and one called the Stokes. And we choose the wavelengths or the frequencies of the pump and Stokes beams so that the difference between them precisely matches a Raman mode that we want to excite. And by doing this, the pump and the Stokes beam interact and they beat together, and the beat between them precisely matches the frequency of the chemical bond that we want to excite. And by doing this, all of the chemical bonds at that specific frequency are coherently driven, which creates a very strong and directional coherent Raman beam. It also has the advantage that it's a non-linear process, and this means that by using very short light pulses, we can enhance the signal even further to boost the signal up to the type of level that we'd expect to get from fluorescence. And there are two types of coherent Raman that we typically use for microscopy. One's called coherent anti-Stokes Raman scattering, or CARS, and the other is stimulated Raman scattering, or SRS. So the two processes are very similar in how they're created. They're both created by shining the pump and the Stokes beam into the sample, but they're detected in slightly different ways. The coherent anti-Stokes Raman scattering is detected as a blue shifted beam that's emitted from the sample. And if you look at the diagram here, you can see the process by which we obtain the blue shifted signal that comes out at the anti-Stokes frequency. This is typically detected using a PMT detector to detect the very small light signal that comes out. Stimulated Raman scattering, on the other hand, is very similar to stimulated emission. What happens when we shine the pump and the Stokes beam onto the sample, by simultaneously shining the photons at these frequencies onto the sample, we stimulate the emission of a photon at the Stokes frequency. And effectively what we're doing is taking the pump beam and converting it into a Stokes photon. So if we were to modulate the Stokes beam whilst keeping the pump beam constant, we would see the modulation transfer from the Stokes beam onto the pump beam as we convert photons from one beam to the other. And we can detect this very small modulation in signal using a lock-in amplifier to pull this modulation signal out from the pump beam. So the way we perform coherent Raman microscopy is we use a microscope that looks very similar to the type of system you'd use for confocal or multiphoton fluorescence. We use galvanometer mirrors to raster scan the focal spot across the sample. And what really works in our favor here is that because it's a non-linear process, the signal's only generated at the very center of the focal spot, and we can scan that spot round in 3D whilst recording the CARS signal with the PMT or the SRS signal with the lock-in amplifier to map the location of bonds within the sample. And in the example here on the right hand side, we can see a video of a 3D stack of data where we're imaging the CH stretch signal at 2850 wave numbers from skin lipids. And you can see that we can map the spatial distribution of the skin lipids with high resolution in the skin up to a few hundred micrometers beneath the skin surface. Remember, we're working with near infrared lasers, so it's less susceptible to scattering than we have with visible light, and it works extremely well in this highly opaque sample. And we've also used it to monitor the diffusion of drugs through the skin. And in the example here, we're seeing an XZ slice through the surface of skin, and we can monitor the diffusion of a drug called ketoprofen, which has a strong peak in the at just below 1600 wave numbers. And we can use this to separate the ketoprophene signal from the skin lipids. And in parallel, we could also use an excipient, or the solvent that was used to transport the ketoprophene through the skin. In this case, it was propylene glycol, and we can map the position of the ketoprophene and the propylene glycol using the skin lipids to provide contrast of the skin structure. So the big question when I first got into this work, I received a phone call from an agrochemical company saying we've seen a lot of work done with coherent Raman in skin. We'd like to see if you could do this in plants. So the problems that are faced by the agrochemical community are very similar to that in terms of the dermal drug delivery guys, in that they want to track the diffusion of low molecular weight compounds through the surface of the plants. So I was rather excited, rushed out in front of the physics building and grabbed a blade of grass, put it in the car's microscope, and was very excited when I saw this beautiful picture of the plant cells within the leaf. But just to check that it was coherent Raman rather than fluorescence, if we block off either the pump or the Stokes beam, we should see the car's signal disappear. However, in the case of the plant, the leaf, what we were to see that if we apply only the pump or only the Stokes, the signal doesn't disappear, and rather disappointingly, all we're seeing is just fluorescence. So if we look on the diagram here of the autofluorescence signal that we get from chloroplasts and plant cell walls at the different wavelengths, we can see that the fluorescent emission is extremely broad, and it completely swamps the small cars signal that's produced from the plant. So the second thing to try was to see if SRS would work. So in principle, SRS isn't affected by fluorescence because we're detecting the signal at the excitation wavelength, not at the redshifted fluorescence wavelength. And if we switch from CARS to SRS, we see a significant improvement in the background. So the fluorescence has now disappeared, and we can make out the plant cell wall structure at the CH stretch. So it looks extremely promising that it's going to work well in plant tissues. However, if we look at these objects here, these very bright signals, these are coming from chloroplasts. And what we decided that this signal was, this isn't fluorescence, this is excited state absorption. So in the presence of the pump and the Stokes being incident on the sample simultaneously, we get this excited state absorption, whereby the pump beam excites up to an excited state that then allows the Stokes beam to excite the system up into a higher state. So when both beams are incident upon the sample, we get a modulation transfer in the same way that we get a modulation transfer from SRS when both beams are incident upon the sample. So although both stimulated Raman and excited state absorption in the plant tissue result in a modulation transfer, there's a little trick that we can exploit in a very subtle difference between the two techniques. Depending on whether we modulate the pump or the Stokes beam, we can choose to detect a loss or a gain in the Raman signal. So stimulated Raman loss is where we modulate the Stokes beam and look for the loss in the pump beam as the photons are transmitted from the pump to the Stokes beam. Whereas if we choose to detect the Raman gain, here we're modulating the pump beam and detecting the gain in the Stokes beam as we modulate the pump beam. However, for excited state absorption, it doesn't matter which beam we choose to modulate. There's no transfer in intensity between the two beams. Both beams are absorbed by the sample in exciting electrons into excited states. So whether we choose to modulate the pump or the Stokes beam, we always see a loss in the other beam. And this is something that we can exploit to separate the Raman, the chemically specific contrast, from the excited state absorption that we want to get rid of. So by using phase-sensitive detection on the lock-in amplifier, we can separate out the stimulated Raman gain from the excited state absorption because they're at a different phase. And we can very effectively use this trick to separate out the Raman signal from the chloroplast signal. So in red here, we're plotting the intensity at one phase on the lock-in amplifier, and we're seeing purely the excited state signal from the electronic contribution from the chloroplast. And in green, we're looking at the stimulated Raman loss, which comes from the vibrationally chemically specific signal from the plant cell walls. So by doing this, we can exclude not only the fluorescence from the highly autofluorescent plant tissue, but we can also exclude the non-chemically specific signal that we get from excited state absorption. The phase-sensitive SRG detection works extremely well in planter, and we've used it to map various different biopolymers within the plant. And by tuning the difference between the pump and the Stokes beam as we acquire images, we can extract the Raman signature from the plants to be able to tell the difference between the different chemical biomolecules within the plant. So effectively, we can replace fluorescent tagging, and we can look at different biopolymers such as pectin and cellulose and lignin within the plant tissue. And very useful for the agrochemical industry, we can look at the wax on the surface of the leaf. So if we look at agrochemical formulations and what we might want to achieve, there are three main areas in which we've been working. We've been looking at the application of agrochemicals on the leaf surface. We've been looking at the application at roots, and we've also been looking at the application onto seeds. And in all of these different areas, what we're trying to achieve is to visualize the movement of the agrochemicals across the various barriers that the plant puts up against the diffusion of molecules. So the cuticle wax is the first barrier that agrochemicals come up against when they hit the surface of the leaf. It's designed to keep water into the plant and to prevent pathogens from entering the plant. But obviously, if you want to deliver agrochemicals across the plant, this is very challenging because your agrochemicals are generally delivered in an aqueous form, and you want your agrochemical to be, on one hand, water soluble so it can move through the plant after it's penetrated the leaf, but also you need it to get through this hydrophobic barrier. So a lot of research goes into understanding the structure of the wax on the plant leaf, and we also want to be able to visualize it so that we can visualize the movement of agrochemicals through the wax with and without various solvents and adjuvants that can be used to enhance the uptake. So we've imaged the deposition of agrochemicals onto the cuticle wax, and again, by tuning into a specific frequency of the agrochemicals, we can map out the deposition of crystals onto the surface. This is very useful because it gives the agrochemical companies an understanding of what the compounds look like as they're deposited onto the surface. The crystal size and the distribution of crystals is very important. But what we really want to understand is how do the chemical physical properties of these active ingredients and any adjuvants or formulation compounds that you mix into the formulation with them, how does that affect how well they cross this hydrophobic barrier? So we've done some work where we've been working with model cuticles. So we've extracted cuticle wax from the surface of leaves and to put them into a diffusion cell whereby we can image a droplet of an agrochemical formulation and take a time series of SRS images separated by approximately 60 seconds, and we can visualize the diffusion of the compounds into the wax in real time. And what we get if we analyze the concentration as a function of depth into the wax is we get a series of diffusion profiles. And what we expect the compounds to move through the wax governed by Fick's law, and which is given by this equation here. And if we plot our data as a function of time in minutes, we can see that the diffusion profile moves through the sample. And if we assume that it does move according to Fick's law, if we recast the data where we divide the x-axis, the z distance, by the square root of the time, what we would expect if it does indeed move with Fick's law is that all of the plots would fall upon each other. But what we can see here is that this doesn't happen. So we can instantly see that we're not seeing a process that's governed by Fick's law. And what in fact is happening is that the diffusion coefficient is changing with time. And this is easier to see on this graph here, where we plot the area under the curve in this graph here, as a function of time. And what we'd expect to see in this case, if the diffusion coefficient was constant, we'd expect all of the points to lie across this curve. Instead, what we see is the diffusion coefficient rising with time, which is showing that the agrochemical formulation is effectively dissolving the wax as it moves through the substrate, increasing the diffusion coefficient, which is what we want to see. And if we repeat the experiment, and this time we add an adjuvant or formulation compound to the formulation, what we see is we can increase the diffusion coefficient by a factor of 2 and also increase the rate at which the diffusion coefficient increases. And this is a very nice analytical tool because it gives the agrochemical industry for the first time something by which they can quantify the effects of the formulation compounds on the movement of agrochemicals through the wax. We've done some work where we've been exploring the size-dependent uptake of agrochemical particles through the roots. And to explore this, we used the excited state absorption signal from gold nanoparticles. So we use the gold nanoparticles as models for particles of agrochemical compounds. And whereas before in the chloroplast we were trying to exclude the excited state absorption, this time we're using the excited state absorption that comes from the plasmon resonance of the gold particles as a marker to show whether we have uptake into the root. So in the green signal, you can see we have the chemically specific stimulated Raman gain signal that comes from the cellulose and the plant cell walls in the root. And in the blue, we have the excited state absorption that comes from aggregated 5 nanometer particles that have aggregated within the roots. And the key issue here is that 5 nanometer particles on their own, isolated particles, do not give us a plasmon resonance at the wavelengths that we're exciting at, around 1 micron. So where we have isolated particles in the solution surrounding the roots or within the roots themselves, we don't see any ESA signal. It's only where the particles become aggregated and form clusters that the surface plasmon resonance shifts to our excitation frequency. And this works very nicely because it allows us to apply small particles that can diffuse through the plant cell walls and show us where they're aggregating within the vasculature in the plant root. And in this case, we're looking at the phloem or xylem within the plant leaf. So we've used this to look at uptake into roots, and we see for 20 nanometer gold particles that were exposed to plant roots for 24 hours. We don't actually see any uptake into the roots. So we do get the excited state signal, but all we see is the signal associated with clusters of particles surrounding the roots on the outside. We don't see anything from the center. Whereas if we compare this to the 5 nanometer gold particles grown under the same conditions, this time we don't see any signal from the outside of the plants. There's no aggregation on the outside. The particles have moved through the cell walls and then have aggregated in the plant vasculature and give us a very strong excited state signal from the center of the roots. So this is a very nice tool where we can control the size of the gold particles to explore what size particles can be efficiently uptaken into the roots and where they travel and aggregate within the root system. So the final area that I want to talk about is seed coatings. So seed coatings are a unique opportunity in agrochemistry to target all of your active ingredients onto the plant without suffering any unwanted collateral exposure to the environment. So the applications that are useful here is that you can coat the seed with compounds that will protect the seed. This could be for storage or to protect them when they're in the ground. So typically we're coating the seeds with fungicides to stop them from being eaten or destroyed by pathogens. But what the agrochemical company want to know is how do the processes by which they apply these actives, how does it affect the uniformity and the coating at the microscopic level on the seed? So in this example, shown on the slide. We're looking at a fungicide called fluoroxyl, and in this case it has this very useful peak with a C triple end bond that gives us a very unique peak at 2219 wave numbers, which sits in the silent region of the Raman spectrum. And we can use this to map out the distribution of crystals on the seed surface to give some feedback to how well their coating processes are working. So we can take a seed and we can compare the formulation of the seed coating, in this case with and without oil. And the oil is added to the seed coating preparation to enhance the adherence of the agrochemical crystals to the surface of the seed. So we have CH2 contrast of the seed structure shown in green. We have the C triple N bond at 2219 wave numbers shown in red, and then we can merge the two and we can see the distribution of crystals on the surface of the seed, and again we can see it in 3D. And we can see that without the oil we seem to have fewer and smaller crystals deposited on the surface of the seed. So it does look like the oil does enhance the coating process. But we can do one better than that. We can take quantitative images. We can image over a large area of the seed coating, and then by using image analysis we can quantify the total surface area of the agrochemical across the surface area of the seed, and we can see that with the oil we have a significant enhancement in the coating of the seed. And we can also compare the crystal size. So with the oil we can see we have a larger population of larger crystals on the surface of the seed. Okay, so to summarize, stimulated Raman scattering is ideal for visualizing the distribution of low molecular weight biomolecules in biological samples. By using the property of stimulated Raman gain versus stimulated Raman loss, we can use phase sensitive detection to detect the chemical signature in the presence of not only strong autofluorescence but also substances that produce strong excited state absorption signals. And from some of the stuff that we've done so far, I think it's starting to look like it could be a very promising tool for development of the next generation of agrochemicals and plant-based technologies. And I hope that you found this interesting, and I've encouraged you all to explore plant-based applications of your biophotonics techniques. So if anyone's interested in finding out a little bit more about stimulated Raman scattering or CARS microscopy, we've recently been awarded the UK's first coherent Raman user facility called Contrast at University of Exeter. It's a fully supported user access facility, and we both have a CARS and stimulated Raman microscope, and also an ultra broadband CARS system that I haven't talked about in this presentation. So we run user access calls 3 times a year. During the COVID crisis, we're not accepting external users, but we are running a postal service whereby users can send us samples and we can image them and send you the data. And if you'd like to find out more about Contrast, please drop me an email. So thank you for your time. Goodbye.